Good morning, my dear students. Welcome to our Zoology online class. Dear students, in the last class we started the fifth lesson, molecular genetics. Okay, we started the fifth lesson. Then in the last class we studied about some important question answers and one important 5-mark question, the properties of genes. Okay, properties of genes is a very important 5-mark question we have completed in the last class. And some 2-mark questions also we studied in the last class. For example, one gene, one enzyme hypothesis, one gene, one polypeptide chain hypothesis. Like that means some important 2-mark questions also we have completed in the last class. Dear students, today we are going to study about 2 more important topics. Okay, so the first topic is on experiment. The name of the experiment is called Hershey and Chase experiment. Hershey and Chase experiment. Listen very carefully, dear students. This question is very important for 5-mark question. See, actually, in the last class I said Griffith. He has done one important experiment, bacterial transformation experiment. Okay, and at the end of this experiment, he said that the DNA is the genetic material of living organisms. Okay, but many scientists not accepted this point or this view of Griffith. Okay, so many scientists not accepted that the DNA is the genetic material. They said they believe that the protein molecules are the genetic material. They believe that the protein molecules are genetic materials. Understand a few? And in eukaryotic chromosome, the amount of DNA and the amount of protein is almost equal. That means in eukaryotic cells consist of equal amount of proteins and nucleic DNA. Okay, proteins and DNAs. Okay, but the protein molecules only consist of some diversities, some chemical diversities and complexities in their nature. So they believe that the protein molecules only can carry the hereditary or only they can the genetic information, carry the genetic information. So these scientists believe that the protein molecules are the genetic materials because they have some genetic diversity and they have some complexity, so they can carry the genetic information from a cell to from a generation to next generation. Okay, they said like that. Okay, here these two scientists, Hershey and Chase, these two scientists togetherly conducted one experiment, and this experiment is one of the best evidence that the DNA is the genetic material. That the DNA is the genetic material. Understand, my dear students? So listen very carefully. So now we are going to study about this Hershey and Chase experiment. Here, actually T2 bacteriophage is a virus. Okay, T2 bacteriophage is a virus. This virus can infect and can destroy the Escherichia coli bacteria. T2 bacteriophage is a virus. This virus can infect or can attack and destroy the Escherichia coli bacteria. Understand? So when these two organisms are mixed together or when these two organisms are kept togetherly, immediately, within few hours, the T2 bacteriophage virus can insert some materials inside the body of the Escherichia coli cell, cells. Understand? And finally, the Escherichia coli bacteria cell is lysis or broken. And from the Escherichia coli bacteria, large number of young T2 bacteriophage viruses are released. Okay, large number of progenies or young T2 bacteriophage viruses are released from the Escherichia coli bacteria cell. Understand a few? Understand? And here, so with the help of this Escherichia coli bacteria and T2 bacteriophage virus, Chase and Hershey, these two people conducted one experiment. Okay, so according to this experiment, the genetic materials, according to this experiment, the T2 bacteriophage having the capacity to destroy the Escherichia coli bacteria. Understand? They observe when these two cells are kept togetherly, they observe some materials are entered inside the Escherichia coli bacteria from the T2 bacteriophage virus. Okay, so they wanted to observe which material is entered inside the bacterial cell from the T2 bacteriophage virus. So, they conducted this experiment with the help of radioactive isotope. 2 radioactive isotopes. One radioactive isotope name is the radioactive isotope of sulfur, and the second one is the radioactive isotope of phosphate. Okay, understand? So listen very carefully, dear students. So now we are going to study about this experiment. So to do this experiment, to conduct this experiment, first they took two culture medium. Just imagine this is one culture medium and this is the another culture medium. Okay, in this culture medium, they allowed some T2 bacteriophage virus. T2 bacteriophage virus. And here also they allowed some T2 bacteriophage virus. Understand? Then in this culture medium, okay, in one culture medium, they added sulfur. Understand? And this culture medium was labeled as protein. Is labeled as protein because this culture medium consists of what type of radioactive isotope? Sulfur. Okay, and in this culture medium they added phosphorus. Okay, phosphorus, and this culture medium is labeled as DNA. This culture medium is labeled as DNA. So listen very carefully, dear friends. They took two culture medium. First culture medium, they kept in the first culture medium they kept some T2 bacteriophage virus, and also in the second culture medium also they kept some T2 bacteriophage virus. In one culture medium they added sulfur and it is labeled out, it is mentioned as protein. And next culture medium they added phosphate and it is mentioned as DNA. Understand of you? Understand? Then now they added a Escherichia coli bacterial cell inside both culture medium. Okay, so unlabeled Escherichia coli bacteria is added in the both culture medium. Understand of you? Now within few hours, these T2 bacteriophage present in the culture medium started to attack or infect the bacteria, Escherichia coli bacteria. These T2 bacteriophage present in the culture growing inside the culture medium is started to infect the Escherichia coli bacteria. Understand? And they clearly observe before the bacterial cells break down or lysis. Soon after the infection, they observe these bacteriophage virus only closely connected or associated with the Escherichia coli bacterial cells. So to infect these Escherichia coli bacterial cells, these bacteriophage virus, T2 bacteriophage virus started to associate or connected with this Escherichia coli bacteria. But here these T2 bacteriophage virus are not connected with this Escherichia coli bacteria. They are present in the surrounding areas. Hope you understand this point. So T2 bacteriophage present in the phosphorus containing culture medium is closely associated or connected with the Escherichia coli bacteria. And here the T2 bacteriophage present in the sulfur containing culture medium is present in the outer surface or the surrounding place. It is not connected with the Escherichia coli bacteria. Understand? So they came to a conclusion. So this T2 bacteriophage only inserted the genetic material inside the Escherichia coli bacteria. Already here what is there? DNA is there. So the T2 bacteriophage present in this culture medium entered or inserted the DNA inside the Escherichia coli bacteria. Understand of you? Then finally they analyzed the progeny or the young ones released from this Escherichia coli bacteria. The young T2 bacteriophage virus released from this Escherichia coli bacteria. Okay, they analyzed this radioactive. They analyzed the radioactive methods to analyze the T2 bacterium or progeny T2 bacteriophage virus. Okay, so that T2 bacteriophage virus also carrying only phosphate. Okay, not sulfur. Understand? So finally they came to a conclusion. Okay, after the analysis of progeny or the young T2 bacteriophage virus, they came to a conclusion DNA is the genetic material, and this DNA only carrying the information from the virus to the bacteriophage virus. From the bacteriophage virus to Escherichia coli bacteria. Understand, dear students? So this is one of the very important 5-mark question. Hershey and Chas experiment. So first they took, so once again repeat, they took two culture medium. In both culture medium, they added the G2 bacteriophage virus. Then in one culture medium, they added one radioactive isotope of sulfur, and in one culture medium, they added the radioactive isotope of phosphate. And this culture medium is labeled as protein, and this is labeled as DNA. Now they added Escherichia coli bacteria in both the culture medium. Here the culture G2 bacteriophage growing in the phosphorus containing culture medium only infect the Escherichia coli bacteria, and from the Escherichia coli bacteria many young or progeny T2 bacteriophage are released. Okay, they analyzed this young or progeny virus, T2 bacteriophage virus. They said the DNA is the genetic material. So T2 bacteriophage inserted the DNA inside the Escherichia coli bacteria. Understand? So this is one of the best evidence that the DNA is the genetic material. Understand, my dear students? Hope you are understood this experiment. So this is one of the very important 5-mark question. Then next we are going to study about the second topic. This is also a very important 5-mark question. Chemistry of nucleic acid. Chemical structure of nucleic acid. Actually, the nucleic acid is a long chain-like structure. Nucleic acids are long chain-like structure. It's made up of many small subunits. Name of the subunit is called nucleotides. Name of the subunit is called nucleotides. Understand? And each and every nucleic acid or nucleic material consists of 3 important parts. One is a pentose sugar, 5-carbon sugar molecule pentose sugar. Second one is the nitrogen base, and third one is the phosphate group. So these are the 3 important components present in a nucleic acid. Listen very carefully, dear students. So the first point I said about this nucleic acid is it is a long chain-like structure. And the second point, it is made up of many subunits. Okay, many repeated subunit. Name of the subunit is called nucleotides, and it consists of 3 important parts. First one is the pentose sugar, second one is the nitrogenous base, and third one is the phosphate group. So first I explain about this pentose sugar. Listen very carefully. Based on the types of sugar molecule, the nucleic acids are classified into 2 types. One is the DNA and the second one is RNA. One is DNA and second one is RNA. If the sugar molecule is deoxyribonuclease sugar, okay, deoxyribose sugar, that nucleic acid is a DNA. If the sugar molecule is a ribose sugar, that nucleic acid is a RNA. Okay, so if a nucleic acid consists of deoxyribose sugar, that nucleic acid is called DNA. If a nucleic acid consists of only ribose sugar, that nucleic acid is called RNA. Okay, only one difference only present between this DNA and RNA. In DNA, one oxygen atom is less than the RNA. Okay, one oxygen atom is less than the RNA. So that's all about this pentose. So based on the types of pentose sugar, the nucleic acids are classified into DNA and RNA. If the nucleic acid consists of deoxyribose sugar, that nucleic acid is called DNA. If the nucleic acid consists of ribose sugar, that nucleic acid is called RNA. The one and only difference between this DNA and RNA is in DNA, one oxygen atom is less. That's all about this pentose sugar. And the second part, nitrogenous base. Listen very carefully. See, the nitrogenous base, based on the nitrogen molecules present in the nucleic acid, there are 2 types of base are there. 2 types of base are there. One is the purine and second one is the pyrimidines. So in each and every nucleic acid, the 2 types of nitrogenous base are there. One is purine and another one is pyrimidines. There are 2 purines are there. One is adenine and the second one is guanine. Okay, adenine and guanine. These 2 are the purines. And they have double carbon nitrogen ring structure. They have double carbon nitrogen ring structure. Understand? And 2 pyrimidines also there. One is thymine and the second one is cytosine. In DNA, listen very carefully. In DNA, thymine and cytosine both are the pyrimidines, and in RNA, cytosine and uracils 2 are the pyrimidines. Okay, so totally 3 pyrimidines are there. In DNA, 2 pyrimidines there. What are they? Thymine and cytosine. In RNA also two pyrimids are there. What are they? Cytosine and uracil. Okay, and these pyrimids are single carbon nitrogen ring structure. Pyrimids are single carbon nitrogen structure. Once I repeat about this nitrogenous base, based on the nitrogen base, there are two types of nitrogen base are present in the DNA and RNA. Or two are purines and totally four nitrogen base, two purines and two pyrimids. Adenine and guanine, these two are the purine. They have double carbon nitrogen ring structure, and pyrimid is thymine, cytosine, uracil. Here thymine and cytosines are the pyrimid. Two pyrimids present in the RNA, and cytosine and uracils are the two pyrimids present in the RNA. Understand? So sorry, thymine and cytosines are present in the DNA, and cytosine and uracils are present in the RNA, and they have single carbon nitrogen ring. Understand, my dear students? So, two important components we have studied about this nucleic acid. One is the pentose sugar, and the second one is the nitrogenous base. Then, next, the third one is phosphate group. Third one is phosphate group. Actually, this phosphate group is derived from an acid. Name of the acid is called phosphoric acid. One more question. Okay, phosphate group of the nucleic acid is derived from phosphoric acid. Okay, and this phosphate group consists of three active OH group. Three active OH group. Among these three active OH group, two active OH groups are involved in the strand formation. Just imagine, you know the structure of the double stranded structure of the double helix structure of the DNA molecule. Okay, two strands are there. Okay, isn't it? So here among these three active OH group, two active OH groups are involved in the strand formation. Active groups are involved in the strand formation. Then the next point is, say, based on the bond present or formed in this phosphate group is ester bond. Okay, the bond formed or produced from this phosphate group is called ester bond. Understand? Then the next point is, after the formation of phosphodiester bond. Okay, after the formation of phosphodiester bond, the oxygen atom present in this phosphate group is negatively charged. Listen very carefully. After the formation of phosphodiester bond, the oxygen atoms present in this phosphate group is negatively charged. Is changed into negatively charged. So this negatively charged phosphodiester bond is used to keep the nucleic acid inside the cell membrane or inside the nuclear membrane. Inside the cell or inside the nuclear membrane. Hope you understood this last point. See, after the formation of phosphodiester bond, the oxygen atoms present in the phosphate group is negatively charged. So this negatively charged phosphate group, phosphodiester bond is used or retain the nucleic acid inside the nuclear membrane or the cells. Understand of you? So that's all about this phosphate group. That's all about this phosphate group. Then next we are going to study about the subunit of the nucleic acid. Already I told you this is made up of nucleic acid. Both RNA and DNA is made up of many subunit. Name of the subunit is called nucleotides. So now we are going to study about the nucleoside and nucleotides. Listen very carefully, my dear students. See, the first point is the nitrogen base. Okay, single nitrogen base is linked with or connected with one sugar molecule. Immediately it is converted into nucleoside. Immediately it is converted into nucleoside. Hope you understood. One sugar molecule is connected or joined with a nitrogen base. Immediately it is converted into nucleoside. Immediately it is converted into nucleoside. Okay. When one phosphate molecule is added with the same sugar molecule, immediately this nucleoside is converted into nucleotide. Nucleoside is converted into nucleotide. So the first points about this nucleotide and nucleoside is when a nitrogen base is connected or joined with one sugar molecule, immediately it is converted into nucleoside. When one phosphate molecule is added with the same sugar molecule, this nucleoside is converted into nucleotide. Hope you understood this first part. Then The second point, say nucleotide molecules. Just imagine this is one nucleotide, one nucleotide, one nucleotide. Okay, so all the nucleotide molecules present in a DNA molecule or RNA molecule is joined by a reaction. Name of the reaction is called condensation reaction. So all the nucleotides are joined by a reaction. This reaction is called condensation reaction. And after many condensation reaction, the nucleotides are togetherly forming. Okay, nucleotides are togetherly forming polynucleotide chain. Polynucleotide chain. Hope you understood this point. So, nucleotide molecules present in a nucleic acid is connected or joined by condensation reaction. Then, after many condensation reaction, the nucleotide molecules, many nucleotide molecules are joined together and forming one chain. Name of the chain is called polynucleotide chain. Polynucleotide chain. Okay. So, 2 points we study about this nucleotide. Nucleotide. Then, next, the third point about this nucleoside and nucleotide is listen very carefully, dear students. See. One nucleotide, the OH group, okay, or the phosphate group present in 3-carbon sugar present on the 3-carbon sugar molecule of one nucleotide is connected with the phosphate group of another one nucleotide. So just imagine, dear students, this is one nucleotide. This is another one nucleotide. This is another one nucleotide. Okay. So the OH group. Okay. This OH group of this is the OH group of this nucleotide. Okay. It is present in 3-carbon sugar place. Okay. 3-carbon sugar. And this OH group is connected with. Okay. Listen very carefully. This OH group is connected with a phosphate group of another one nucleotide by S bond. By S bond. Once again repeat this point. So the OH group present in the 3-carbon sugar molecules of one nucleotide is connected with the phosphate of another one nucleotide molecules by a bond called S bond. Okay. Then the next point. The sugar molecules present in 2 adjacent nucleotides. So this is one nucleotide, this is another one nucleotide. So the sugar molecules present in 2 adjacent nucleotides are connected by a bond. Name of the bond is called phosphodiester bond. Okay, phosphodiester bond. Listen very carefully, dear students, this third point. So the OH group present in the 3-carbon sugar molecules of one nucleotide is connected with the phosphate group of another one nucleotide by a bond called X bond. The sugar molecules present in 2 nearest or adjacent nucleotides are connected with the phosphodiester bond. So this is the third point about this nucleoside and nucleotide. Understand a few? Then the fourth point. Both the DNA and RNA has 2 ends. Okay, just imagine this is a DNA. This is a DNA, and this end is called 5 symbol end, and this end is called 3 symbol end. Okay, one end is called a 5 symbol end, and the second end is called a 3 symbol end. Okay, and in both end carbon is present. In both end carbon is present. Okay, then in 5 end, okay, 5 symbol end, in this 5 symbol end, one phosphate group is attached. One phosphate group is attached. Okay, one phosphate group is attached. And in the C carbon end, one OH group is attached. One OH group is attached. So, hope you understood. In DNA and RNA, both nucleic acids consist of 2 ends. One end is called 5 symbol end and the second end is called 3 symbol end. In 5 symbol end, one phosphate group is there and in the is added, and the 3 symbol end, one hydroxyl group is or OH group is added. It is in DNA molecule. Okay, and in a RNA molecule, just imagine it's a RNA. Here also two ends are there. One is the 5 symbol end and the second end is the 3 symbol end. Okay, so here also carbon is there. This carbon is attached with the phosphate and this carbon is connected with one OH group. Okay, but in RNA, one more additional OH group is present. One more additional OH group is present in the second position. So just to imagine this area is the second position area. So one and only difference between these two DNA and RNA is in RNA one OH group is additional present. Additional OH group is present in the second position. Present in the second position. So dear students, about this nucleoside and nucleotides we have completed 1, 2, 3, 4 points. 4 points we have completed about this nucleotide and nucleoside. Understand a few? Understand? Then the next point is, see, Wilkins and Franklin. Okay, Wilkins and Franklin. These two scientists togetherly analyze the chemical nature of the nucleic acid by using X-rays. By using X-rays. So it's a 1 mark question. Okay, so Wilkins and Franklin, these two people togetherly analyze the chemical structure of the nucleic acids by using X-rays. Then, in the year of 1953, Watson and Crick, these two people togetherly introduced double helix model of DNA. Double helix model, you know. Okay, this is the double helix model. Okay, so in the year of 1953, Watson and Crick, these two people togetherly discovered this double helix model of the DNA molecule. Okay, then another one scientist, Erwin Charcraft. This person saying always the adenine is linked with thymine by 2 hydrogen bond. Always he observed, okay, Erwin Charcraft observed that adenine is always linked with thymine with the help of 2 hydrogen bond. Okay, then guanine is always linked with cytosine with the help of 3 hydrogen bond. Guanine is always linked with or pairing with cytosine with the help of 3 hydrogen bond. Okay, then the ratio between the adenine and thymine and the ratio between the guanine and cytosine is always constant and equal. If 10 guanine is there means actually 10 cytosine is also there. If 20 adenine is there, 20 thymine is there. So the ratio between the adenine and thymine and the ratio between the guanine and cytosine is always constant and equal. Understand? You? Then, if we know the nitrogen base of one strand, we can easily predict the nitrogen base present in the second strand. Just imagine, so this is one strand, this is second strand. If you know the nitrogenous base present in this strand, we can easily predict the nitrogenous base present in the second strand. Understand a few? Dear students, I repeat the last some points now. Some scientist names we have studied now. The first scientist name is Wilkins and Franklin. They only first analyzed the chemical nature of the DNA or nucleic acids by using X-rays. Then second 1 mark question, Watson and Crick. In the year of 1953, Watson and Crick, they only togetherly introduced or discovered the double helix model of the DNA molecule. Okay, and the last scientist name is Erwin Charcraft. Okay, he only first observed adenine is always pairing with thymine with the help of 2 hydrogen bond. Guanine is always pairing with cytosine with the help of 3 hydrogen bond. Okay, and the ratio between these two nitrogenous bases is always equal. Nitrogenous base is always equal and constant. Okay, dear students. So, dear students, today we have completed 2 important 5 mark questions. Well, for the first 5 mark question is only experiment, Hershey Charles experiment, and the second one is chemistry of nucleic acids or the chemical structure of nucleic acids. Okay, both are important for 5-mark questions and many 1-mark questions also we have studied. For example, who discovered the double helix model of the DNA? Watson and Crick. Okay, in which year they discovered the double helix structure of DNA? 1953. Like that, many 1-mark questions also there. Okay, dear students, try to complete today itself, and we can meet in the next class. Thank you.